Well, it ends up with like low energy, but it's still gonna be more than I started with. I am. No, I just my hardest part is seeing you suffer through immunology. It's not that I
seeing you suffer.
I don't have a problem with it too much. I mean, my voice is starting to go. Actually, I need some water. You can read the first news line.
Just read it.
Well, you want to fill in? I'm up there.
Oh, be the front?
You know it. Take pass for 10 minutes.
Yeah, I talked about that. I'm winning.
So what's the deal?
You win. I win.
You read it?
I'm ready.
Next one.
Read.
I already lost.
So we went through this already. We'll go back to the cooperation, but between immune cells. But so the white blood cells that have receptors for antigens are binding these antigens, and then there's an immune response. And we already talked about that in lymph nodes and in the spleen and other areas in the body, and you can look where that is in some of the pictures where you look at the lymph system. That's where the antigens and the blood cells, the white blood cells, are concentrated, and they come into contact with each other in those areas. Lymph nodes, spleen, etc. So the antigens aren't just recognized, but they have to be processed and then presented. And always remember, if you go from first line, second line, third line, there's a lot of communication between cells, but this communication is different. So this is the one where you actually not just send a signal, a chemical out, that then creates chemotaxis, or through the process of chemotaxis attracts cells to it. But this one is a real interaction, a physical interaction between B and T cells. And so doesn't have to necessarily be a B cell, so it's the antigen presenting cells. And there were macrophages, then what do they call it, dendritic cells, and the B cells. So so these antigen presenting cells are able to modify the antigen, then along with their receptor, with their MHC2 receptor, bring it to the surface and present it to T helper cells. So there's a direct collaboration. That's what I want you to remember. There's a B cell or a macrophage or a dendritic cell. They internalize, so they're able to modify the antigen, whatever needs to be reacted against, and present it to T helper cells. So those are the. Does anybody remember what the CD4 or 8 that the CD helper cells have? So they have a CD4. That should almost be an A for the day. And so there's interleukins, and I'm not just another another molecule that's secreted and activates cells, but I don't think if we come back to that too much, and I don't want to make or confuse with too many molecules and processes. But so if you look at, and it should be in your book, if you look at what happens, you have this antigen presenting cell, one of the three cell types, and it basically internalizes. It has receptors. It can actually internalize this microbe, and then what it does is it brings the antigen, it processes it, and it brings this antigen up along with the MHC2 receptor. Remember, those are the only ones that have this MHC2. All the other cells have MHC1. They have an additional MHC2, and they present that at the surface. And then it's presented at the surface, and in addition to that, there's this interleukin, this chemical. And what happens is when they present that to a T helper cell, which is the green one, then the T helper cell can respond, and the interaction between the antigen presenting cell and the T helper cell is, as I said before, so the CD4, which is the one from the T cell, and then the MHC2, those two receptors have to be able to lock to each other. And also, in addition to that, so you have that the T cell receptor next to that CD4. So basically, the antigen presenting cell is presenting whatever is the antigen on its surface, or in that variable, binds it to that variable structure, and that connects to the T cell receptor. And then in addition, that CD4 has to attach. So it's just an additional binding mechanism that has to happen to initiate a response in these T cells. And then the activated T cell assists other cells. So basically now we have a T helper cell activated. We can just go that far, because later on we'll learn what that means, but it then creates a response in B cells again, etc., etc. But so the main message here is that the antigen presenting cells, there's only 3 types. They have MHC2, they process whatever is the antigen, they show it at the surface, and then T helper cells can recognize that, in addition, bind with the CD4 and be activated. So it's a process to activate, to signal to T helper cells, I have found a foreign molecule, and then the activated T cell will create a response in other cells. And we talk about those responses in a little bit. So one thing I already talked about. So if you look at what the B cells do, in addition to processing the antigen and interacting with the T helper cells, those B cells are also stimulated, or a process is initiated to create clonal expansion. So the B cell that recognizes an antigen is also now, after presenting it to the T cells, is going through this rapid mitotic division to produce lots of clones. So what that does is, if you have a B cell that has an antigen bound to it, it does 2 things after it showed it to the T cells, and that's important. So if you have a B cell that's activated by these processes, the division of that activated B cell can give rise to plasma cells, and they produce antibodies. So now we have antibodies, and everybody knows from regular science, or from even just being sick, antibodies is what in the end is the protein that's produced against a specific antigen. So now that you have one cell activated, it shows it to the T helper cell, says, this is what we're working against, and then then these clones produce, and they can make antibodies, which you need against a specific antigen, and it also initiates the creation of memory cells. So there's a part of the cells, a part of the clones is memorizing what they were just working against or working with, and a part of the clones is producing antibodies really quick. So those antibodies can immediately go into action, and
And those are all coming from the B cells.
they're what?
They're coming from the B cells.
Yeah. So the B cells, so you have one B cell, that one B cell that has the right receptor. Remember that? Like it's this one, there's one out of millions that has the right receptor for an antigen. Binds it, shows it to the T cell, but also gets activated. And then after it shows it to the B cell, it produces tons of clones. So it just produces millions of cells of the same type. And some of them make antibody, which is something we are interested in. You can inject antibody against something, and that goes into immunization. Like if I know you produce an antibody already, I could try to figure out how I can get that from you, so I don't have to do it myself. And also memory cells. So it produces memory cells in addition to that. And that's for later, that if this antigen is encountered again by your body, you already have cells that recognize it. And these antibodies that we're talking about are also immunoglobulins. So they're just like that receptor. They have this Y shape. They have, in this case, they have a the constant heavy chain, or the constant part of the heavy chain, and then the variable one here, and they call those the lower part the Fc part, and then the Fab. But really important is just that they look the same, that they can bind the antigen. So once these antibodies from the B cells are in your bloodstream and in your body, they can basically, because of this, because of the binding site for cells here, this end binds the antigen. So the free-floating antigens, or the microbes that are in your body, are bound here, and then this one can attach to cells, and then these cells can, for example, internalize the whole thing or destroy the antigen. So it's just antibodies are free in your body immediately, initiated by this first encounter. B cells produce these these immunoglobulins that are specific against an antigen, and the structure is similar to what we've seen with the receptors. I mean, that was an immunoglobulin. It was immunoglobulin G, I think. There's different ones. I'll show you which ones there are. So these then can, once they're out there binding antigen, they can be bound on this site to immune system cells. And so it just shows here that this antigen binding region can be a good fit or not so good, or a poor fit, depending on how good this antigen is produced, how well this antibody is produced against a specific antigen. So they're not always perfect fit. But that's a side note. So the main interaction then is, now you have an antigen that that was recognized by a B cell, shown to a T cell, and then created this production of antibodies. What happens with those antibodies? And it shows these different 6 processes. We can go through them quickly. So the antibody helps to, and it says here, to either call attention to this antigen, to neutralize the antigen. And so what it does here is, if you look, for example, at so in this case, for example, the antibodies that are produced by the B cells bind to the bacterial cell. That's one possibility. Opsonization is coating microorganisms, other particles with these antibodies, because a lot of times these antibodies are much smaller than the microorganisms, so they're just attaching to it. They produce against a specific structure, and now they attach to the structure and basically make this cell or attach to this cell. And with that, phagocytes like the macrophages are able to recognize it better. It's a little bit like the complement system, and I think that comes in here somewhere, where you have a protein. Just the difference is the complement is a blood protein that just recognizes some structure on microorganisms. Here you have a very specific antibody that was produced by a B cell after it internalized part of that bacterial cell, for example, and so it's a very specific antibody binding to those, and so those organisms are then coded and can then be recognized by macrophages. In viral cases, for example, it can neutralize the virus. We talked about we have a few examples what the body does with viral infections, and in this case, you would basically have antibodies that recognize a specific structure on your virus, and then if it attaches to these structures, it basically, the virus has no more way to attach to a cell and infiltrate it. Remember how those were important? The receptors on viruses were enabling the virus to enter the cell. So now that you have antibodies against that virus, they're covering all these structures, and the virus cannot infiltrate the cells that it has specific receptors for. So it can be attached to it, tagged with it, opsonization, and then neutralization. It neutralizes these virus particles. So maybe the main thing about antibodies is they don't like the complement. They don't put a hole into the microorganism. They basically neutralize it. They tag it to make it more visible, or to make it more important for the immune system to react to. The other thing they do is agglutination. So it means they bind these microorganisms together. They cross-link bacterial cells, which means they're not able to move around. And then complement fixation. So that's where the complement comes in. It's part of activating the complement pathway, and then these cells are actually able to get lysed by the complement. So that's where complement and antibodies come together. So you would have antibodies initiating the process of complement attachment, and then making these. Remember these channels that the complement was able to produce. So that way a bacterial cell can actually be lysed. And then precipitation would mean you have smaller molecules, antigenic or antigen molecules that are bound, and so they wouldn't be free anymore. The antibodies would bind to them. So just keep in mind that mostly the antibodies that are produced by B cells create these different protections. They alert the rest of the immune system to those, so that they can recognize them, or they neutralize them, or they prevent them from entering cells by linking them together. They can activate the complement and lyse them. That's the only way they actually destroy them directly, but it's done by the complement system, or they precipitate antigen molecules out. So if you would get a vaccination that consists of antibodies that are produced by some other human or animal or other system, then that would be part of what happens, and you would get a specific antibody or antibody from this host, and it would apply in your body directly. It would do all these things in your body directly, all those, and it would do it really quickly. So you wouldn't have to go through the steps of B cell internalizing, showing it to T cells, producing clones, producing antibodies. The antibody is already there. The final product gets into your body, and it immediately does these things. And if you know you're suffering from a specific virus, and you know that vaccine is out there, then you can give that antibody, and it immediately recognizes and neutralizes it, for example. So it's a very quick way of helping your immune system out. Not going to go there, that's just part of that. So these are and I'll show you a picture first, these are the classes. So those are the different immunoglobulins. There's IgG, IgA, IgM, D, and E. So those are the 5 different groups you have, and they're different sizes. Just so you've heard them, M is the biggest one. But those are the different types of immunoglobulins that we produce, and then they bind to with this. So they bind. What do they bind here to? So they bind to antigens up here, and then up here they bind to cells. And you can see, for example, that IgG binds to phagocytes. They bind to other cells like mast cells or basophils, and then create. So those are more for against eukaryotic cells. So they bind to specific cells that then produce a response.
The M1s don't bind to anything?
The M1s, they sometimes, if they're just bound to. If they're just, I think what happens is either they bind to a specific cell, or they for example, produce a response, like in this case, and then they also, I guess, serve as a B cell receptor. So that's actually the class I was talking about, I think, initially. So they have a different. How do I explain that best? They're not just antibodies that are in your body, but remember the immunoglobulin, that's actually a receptor. So that's the role of IgM. In this case, it can serve as a B cell receptor. This is the most common one. I remember that it says here 80% immunoglobulin G. That neutralizes toxins, viruses, etc. So that's a really important one, with 80% in your serum, and binds to phagocytes. So that one is able to do a lot of the work. So if you have if your body's reacting to a infection, you most likely have a lot of immunoglobulin G in your serum, and that's specific to whatever you're dealing with, and so you find a lot of that, and that would probably take care of most of the antigens that you're dealing with. Usually what you see is, and that's also not so important, but what's important here is it takes some time with all the recognizing and presenting and then producing antibodies. If you look at what they call the antibody titer, So it means the concentration of antibody in your blood after you're exposed to something, to some antigen, it takes up to 12, 13 days for that response, and then you find more immunoglobulin M and then the IgG that you usually that's the main one working against your disease, against the invader after between maybe 2 weeks and a month, and you have like a increase in concentration of that. And then if you look at in comparison to a secondary response, you have the secondary exposure. So now these B cells that are memorizing this antigen, they can come in right away, and so a day, 2 days after, you immediately have a huge amount of immunoglobulin G produced in your body, and so you can right away have a good immune response to whatever you've been exposed to initially. It's a rapid and a strong response the second time around. This one is maybe interesting for some of you because it's some of the more interesting work that they do. So if you would like to have, it makes sense to look for antibodies against all kinds of different disease, and you would the problem is in general that if you look at a person and you look at their antibody titers, you would usually find a mix of a lot of different antibodies, because we're dealing with so many things at the same time. I mean, we're always dealing with pathogens that enter our body. We're dealing with many of those. We're dealing with other antigens. So you have a mix of those. And the solution, if we could produce an antibody that's basically specific for one antigen, and we say this is the one we really want, then we'd have a solution for one specific antigen. So you could say if there's a tumor cell that expresses a certain molecule or a certain receptor. So you have a tumor cell that you want to attack, and you would like a specific antibody, then it had to be monoclonal, meaning you don't just give a whole bunch. You have an antibody that's specifically produced against that cancer cell. And so what they do is they inject whatever they want a response to, whatever they want to produce antibodies for. They inject that into a mouse, and then they isolate the activated cells. So the mouse does exactly what we do. They produce, I mean, it's an immune system that's not unique just to us, but what they do is they have an activation happening by B cells. T cells are presented with that, and they start producing antibody. And then if you take one of those activated cells, so you have a B cell that's activated, and you have to find out which, so if you present the mouse body, that's probably the best way to do it. If you present the mouse body with a high percentage of an antigen, it's likely that the activated B cells that you get in the mouse it's likely that they're specific against this antigen. So all of a sudden you have a lot of activated B cells, and then if you fuse them with a myeloma cell, which is a blood cancer, which means that cell has no restrictions in terms of how it grows. So you have this cell that's a blood cell, and you fuse it together with this antibody producing cell, then all of a sudden you have a single clone with a single specificity, I mean when you select for that, and it's also producing without end. So it's just producing antibody And so what you can do is then you put these cells that you fuse and you put them into cell culture, which means you give just like we do for microbes, you give the cells, these B cells, good nutrition, good environment, and they produce and they produce and they produce. And they produce antibody, and then what you can do is you can basically, now that you know you have an antibody, that you have a massive antibody that's specific for something. So say you initially said, I want an antibody against a specific cell, cancer cell, and you produce antibodies for it, you connect them, you hybridize them with something, with toxins, for example. So you now have you have antibodies that get into your body, and they're immediately looking for those cells. They're specific for those cancer cells. They move to those cancer cells, and they're attached to toxins, and those toxins kill the cancer cells. So there's a lot of, it's almost, I don't know how to compare it, but there's a lot of research going on to make that more efficient and to make it work better. But it's a smart way to say, if we can go in and fix this, our immune system is trained to fix specific problems. So we'll just use the immune system of some other animal and have it find those cells. So that would be where monoclonal antibodies come into therapy. And then if you look, there's a lot of drugs that are already using these systems. So used in therapy for breast cancer, lymphomas, other cancer cells. So this is where monoclonal, and that's monoclonal antibodies come in, and you can, if you look at the drug, usually it says MAB at the end, mAb for monoclonal antibody, MAB, so that they add that to the name, and so you could recognize it by that, and say, okay, there's specific medications out there that are basically antibodies connected to a toxin that were produced in cell culture or in other animals. So the last thing we have to do really for this is to look at what the T cells do. So now we've covered the B cells. They're able to recognize antigens by their huge variety of receptors, and once they have these antigens in them, they can present those to T cells. Now we'll find out what they do with it. But the B cells themselves produce millions of the same one specific for that antigen, and they make antibodies that then do all these other things with the microorganisms, or they produce memory cells. And so what do the T cells do after they've been alerted by the B cell that there's a certain antigen out there? So we already looked at the T helper cells, and they have CD4 receptors, and then the T cells, the cytotoxic, I guess, I don't know if they're called TC, but they have CD8, so they have a different receptor. So when the T cells are presented, they can act directly against the antigen. They can also secrete cytokines, so they again send signals out to other cells. And they also produce long-lasting memory T cells. And it's just basically the T cell system that does a little bit different things than the B cell system. They're not producing antibodies, but they also have, they either act directly against, so the cytotoxic T cells would act against the antigens immediately, or they signal other cells, or they produce memory cells. So not that different from B cells, except they're not producing antibodies. So the T helper cells, yeah, they call them cytotoxic TC. So you have helper cells 1, helper cells 2, and the cytotoxic cells. So those helper cells usually signal by secreting all these different factors, interleukins, tumor necrosis factor. They're such as interferons. They send those out to activate other cells. And then the helper cells, the Th2, they do the same thing just for other cells, but you couldn't combine them. I don't care if you know T helper cells 1 and 2. Just there's helper cells and there's cytotoxic T cells. And the cytotoxic ones, they're toxic, so they destroy foreign cells. The helper cells help by sending signals out. The toxic cytotoxic cells kill. That's all you need to remember.
Perfect.
That's not the end.
That's not the end.
What? That's not the end. Oh no, that is the end for this. So cytotoxic T cells, and there's some we talked about these. So just that they're involving other macrophages. They're the ones who signal to other cells. The cytotoxic T cells destroy other cells. And then natural killer cells are a type of T cell, but just keep that in mind. Don't worry about anything else because they're not as they might be important, but they're not. They're somewhere in between, and there's a lot of cells that all you have to know, or you already know. So this is the basic for the, and that's just going through the process one more time, nothing new to learn, just that you have these presenting cells, the antigen presenting cells, and you remember what those are. They presented to those CD4 cells, so there's this connection we looked at earlier between antigen presenting and the receptor and the CD4. So what they do then is they produce interleukins, so they activate B cells, they activate macrophages. So this is the helper cell activates, and then the CD8 receptor cells, the cytotoxic cells, actually kill the cell. So that's really all you need to remember from this picture is that those T cells either signal or destroy, which they can do both. They don't produce antibody like the different T cells. And just like the B cells, they also produce memory T cells. So they do produce these cytotoxic memory cells, and they do produce these signaling T cells, signaling helper cells. So if the body encounters that again, it will have B cells that memorize the antigen, and so there's going to be antibody production, but there's also going to be T cells immediately ready, these ones that can destroy those cells, or some that recognize it and immediately produce or activate other cells. So that's why the secondary reaction is so fast.
Cytotoxic.
Yeah, so any lymphocyte that finding and destroying cancerous cells is most likely, because the B cells wouldn't do that. They have to be activated and they produce antibodies first, etc. These ones are kind of like this, and another one, T helper cells wouldn't, so C. So we can go through this pretty quick because this is very intuitive, I would say. So 10 more minutes maybe, and then you're first of all you're out of here. You only have one class left, and this is easy. I mean, this makes sense much more than all these different terms. And why are T cells and B cells producing memory cells? I don't know. They just decided to do that. It probably makes a lot of sense, and it's really like you go back to the beginning and say, how do we deal with all these microorganisms? And that's how they do it. We just have a really impressive system to keep us healthy the whole time. So this is the final. It's about immunity. I like these straightened out. I got some issues. And it makes sense that you have, so active immunity means your immune system is actively producing these cells. It's actively dealing with an antigen. Passive means it's donated by another individual. And so it really is the same thing. Natural immunity means you're actively creating immunity by being exposed to things. Artificial means you're not exposed to it naturally, you're artificially exposed to antigens by, for example, getting it from other people. So essentially, this relates to this, and this to this, and the next slide shows you. There's just, as always, there's more ways to look at it, but you have the natural immunity and the artificial, and the natural would mean you get sick, you get an infection, just focus on those bottom ones. You got an infection, your body's working against whatever is in there, and it does all these steps that we just went through. It's the first line of defense, the second, first meaning, you say your the air that comes in, so because you have an infection, the first one didn't work, that's for sure. And then the second, there's these processes have worked, and most likely your third line of defense is already working on it. So you get an infection, you produce immunity. Passive in this natural way is by getting it from your mother. So you're basically acquiring immunity by, for example, nursing. And there's not a lot of antibodies, I think, that can actually cross the placenta. There's maybe one, but most of the things you're exposed to is going to be through the not exposed to, but the immunity you acquire naturally comes from your mother breastfeeding. And then artificially means you get a vaccination. So your immune system is not naturally exposed to it. It's an artificial an injection, an artificial exposure to an antigen, and that can be active in the way that some antigen is introduced to your body, and your immune system thinks, oh, this is naturally occurring. I'm going to have to work to make this go away. They don't know you injected it, or they wonder why you injected it because they're working so hard not to get anything in, and then you inject it. And then the passive one would be, like we talked about, you don't even have you're really lazy, and instead of at least having your own body produce antibodies against things that you inject them, you say, just give me the final product, which is antibodies. So you're passive. It's artificial and it's passive. And so you would get antibodies and immunotherapy from somebody else. But there's nothing wrong with being lazy in this respect. So these are just a couple examples for each. As we said, so this would basically be an example for this part here. You get antibodies from that are produced for you. Immune serum globulins are usually what you get, and it's part of immunotherapy. You can treat infections or diseases like measles, hepatitis A. It can be produced in horses, but then sometimes you have a reaction against some of the sera from horses, so it's not always a good idea. And it acts immediately. We talked about that because everything is done. You get the final product attaches and does everything it usually does. So that's the lazy immunity immune system choice.
So is this normally for speed, or because your immune system isn't working properly, or why would you normally do that?
Yeah, it could be. It says, I think here, replacing antibodies in immunodeficient patients is a possibility. It would be, yeah, for speed. So if you think back to, which is a different case, but if you think back to the difference it makes to the 4 weeks where the virus moves into the brain versus, so you have to be quick, and it's an immediate solution. I don't know how often that immunotherapy is applied. So this would be the artificial one. That's vaccination. We see that all the time. You get your immune system gets activated by exposing you to an antigenic or an antigen, and there's different ways to do that. The good thing here is you don't just get the final product. Your immune system is working, and so next time you have a powerful and sustained. Wondering about those influenza virus, influenza vaccines, for example. If you would 1 year get it, and then I don't know how long they stay. What happens if they don't really get it right, but then the next year you're already ready for the one because the virus mutated back to the form that they gave you? Is that possible?
Sure. Why not? Yeah.
I don't know. I'm asking the question.
No, I think it is reasonable.
Yeah?
I don't think it's good.
No? Because you're. Yeah. So you mean the mutation part? You would say the memory cells are still there, but the mutation would be, it would be strange to have an organism mutate back to a form. Yeah, that could be. I don't know.
You're about to get hit.
Maybe over more generations of that virus, but yeah, in terms of evolution, it doesn't make a whole lot of sense for the virus to go back. Did you say that too?
No.
Oh. I thought we agreed. So there's different ways to do that. You can kill a whole cell or inactivate a virus. Obviously, you can actually introduce live cells of viruses, a little bit more dangerous, but they're usually changed to a point where, for example, that they're not able to live at your body temperature. That's a change. Or you take some gene out that is the most toxic, produces the most toxic parts, so you can actually do live viruses in cells. You can just use antigenic molecules, so you just use small parts of it, or you genetically engineer them. There's nothing you can't do with genetic engineering, so that's always the point. Clearly, always a big point is if you want to have a good vaccine, it should be inexpensive. We've done that before. We always find inexpensive at the bottom. But it's the same thing. It shouldn't, where was it that you had to learn? Where did you have to write that down? A chemical or physical treatment method, was that it? Or what was it where we had, shouldn't harm the host? Say what?
Ideal drug.
Drugs. So same thing for vaccines. You don't want them to.
Hey.
You don't want them to fall back into. First of all, they should work for you. They shouldn't harm you, and at the same time, they should be working against whatever you encounter, which doesn't always work, as we know from influenza vaccines. Long effects should not require a lot of boosters. You probably had to go for those before too. And then that's just showing in the last couple of minutes how that works. So if you inactivate or kill a virus or a virus or microorganisms, say through heat or through chemicals, it's dead, but it's not working, but it still has the antigens that your body needs to produce immunity. So it still has the structural integrity or part of the structure that you need to produce an effective response. And then if you use a live one, as I said, you can remove, for example, the virulence gene. So then you have a live organism in there that produces probably a stronger response. The one problem here is that, you know, I guess there's always a chance that you actually inject or are introduced to a microorganism that can actually be pathogenic eventually, because maybe it mutates back in your body to it, which I think is a possibility in that case. Yeah. And that's the disadvantage, though. I guess can mutate back. And then finally, the non-cellular, the acellular vaccine. So you just use a specific part of it, and that antigen is then producing antibody or is initiating the response just from a say a surface protein, exotoxin capsule. Makes sense. You don't use the whole cell. You don't need you don't use a live organism. You just use a part. And then genetically engineered. We've all looked at plasmids before. You all transformed bacteria with plasmids successfully to do the same thing and include in the plasmid a surface. So basically, it codes for antigen, and then your own body produces it, and then cells can produce antibodies against those antigens. That's actually something they experiment more with now, which would mean they make some of your cells produce part of a toxin or part of a microorganism, and then your own in your body, and then your immune system is going to produce antibodies against that. So that's one other option. I think it's mostly they're looking at that. It's experimental. Those experimental vaccines for AIDS, herpes simplex virus, leprosy, tuberculosis. So it seems really a really good method, and they've tried it out in animals, but in experimental animals, but haven't tried it in humans. Same thing, right? DNA, plasmid, into the cell, done. See you later.
Bye.
Take that home.